Our first speaker this morning is Sean Kevlahan, who is Chief Executive Officer of Quad Technologies, and he'll be talking about new tools to improve cell viability, purity, and recovery. Sean?
Well, thank you, Ann, for the great introduction, and thanks BPI for inviting me to give you a little talk and give you a little color on Quad Technologies. So a little bit about Quad. So we're an early stage company founded in 2013, and really we take a novel approach to the cell and gene therapy market and offering bioprocessing tools with a biomaterials focused. We're headquartered in Woburn, Massachusetts, just about 15 minutes north of Logan Airport in Boston, and we're operating off a platform that we call QuickGel. And it's a hydrogel-based technology that we can tune to actually make varying sizes of hydrogel microbeads that can be dissolved under gentle conditions. We focus products on mag clouds and clouds, that's with magnetic beads and without magnetic beads respectively, for research and bioprocessing applications, providing customers a gentle and scalable solution for cell separation and even T-cell activation and expansion. Our focus has been with the CAR-T and TCR folks, helping improve and streamline some of their workflow challenges. And we're developing cGMP capabilities to support clinical grade products in the clinic. So it's a really exciting time right now because within cell and gene therapy, there is some remarkable findings in terms of complete responses for select leukemias. Some of you might have heard it, CAR-T and TCR might ring a bell. So some people might be familiar with CAR-T and TCR, and they've been sustaining some really nice durable responses, greater than 50% CR within select leukemias, including non-Hodgkin's lymphoma and other various leukemias. And so this is truly the next paradigm in therapies today. But there's a huge challenge, and that challenge is that we're dealing with a cell therapy product, and that cell therapy product, you need to actually address that those manufacturing issues in a very different way. And so we address that through biomaterials. And so as I mentioned before, we have a platform technology that we call QuickGel that allows for a capture and release of cells from substrates without introducing any deleterious agents, such as enzymes or competitive binding, that allows you to streamline your workflow much easier. And so we're focused on applying QuickGel into two distinct unit operations, first being T-cell selection, and the second being T-cell activation and expansion. Now, QuickGel is really a remarkable polymer because it has a variety of different applications within cell separation, but as I mentioned before, our value is that we can gently disassociate this polymer with ease. So within 5 seconds, we can nearly instantaneously decouple cells from substrates. Now we can make these particles in a highly tunable fashion, ranging, these particles can be made as low as 5 microns to as high as 500 microns, depending on what application that within a cell therapy manufacturing process is needed. Now we have standard conjugation chemistries that we can actually derivatize capture moieties to purify T cells, as well as signaling moieties to the surface to activate and expand T cells for CAR-T and TCR. And at the end of the process, we offer, again, a magnetic bead-free solution for T-cell selection, but is also activation, actually eliminating unit operation steps. So let me talk a little bit about what that manufacturing process looks like. So conventional CAR-T and TCR manufacturing, whether it's autologous or allogeneic, starts with a complex suspension of apheresis that's sourced from either healthy or disease-burdened patients. And we implement our technology within 3 distinct unit operations. The first being T-cell selection, that's purifying T-cells from that apheresis material, and then activation and expansion of those T-cells, allowing us to expand those T-cells prior to gene transfer or training those T-cells for that oncology indication, and then expanding those T-cells further for formulation prior to cryopreservation and then actually being dosed back into a patient. So we took two distinct tracks. The first track we took is towards cell separation. And so we developed a product that we call MagClouds. And MagClouds is our polymer impregnated with magnetic particles, so you can manipulate these quick gel particles in a bioprocess or a cell separation experiment. And what we've done is actually we've derivatized our MagClouds particles with streptavidin, so one can use a biotinylated antibody to do their cell separation experiment. And at the end of the process, you can decouple those magnetic beads. Now, in the other tract, we're looking at T-cell activation and expansion, which is the second unit operation within CAR-T and TCR. And what that does is allows these T-cells to actually activate, expand, and then begin to differentiate, so you get more and more of these CAR-T cells in culture. So let me touch upon MagClouds first. So conventionally, people do cell separation with magnetic particles. This is a great academic tool, but has been difficultly translating into a bioprocessing tool. And those steps are pretty intuitive. So you first bind those magnetic beads specifically to your T cells, and then at the end of the process, you actually have to remove those magnetic beads. But the process of that removing those magnetic beads, because they pose a contamination issue and a tox, that you actually lose about 30% of your product downstream. So our thoughts were is to apply our proprietary polymer. Once again, it's acting as an intermediate between the magnetic particles and the cells, allowing you to disassociate those magnetic particles with ease, thus clearing those magnetic particles without any product loss. And this really paints on why releasable chemistries are important, because more specifically in a very expensive process like CAR-T and TCR, you're not suffering from a 20 to 30% loss downstream when you're dealing with conventional technologies. But with a releasable technology, you're not only decreasing the amount of residual magnetic beads, but you're also not actually having that recovery hit that you would have downstream. Now, in addition to that, we allow for even more flexibility in sorting. So the ability to positively select subsets of T cells within process allows you to really develop workflows that are much more robust, and that really can't be done today with conventional magnetic bead technologies. So let me show you some data that we've gotten for cell separation. So within cell separation, again, we've been focusing on pretty exclusively CAR-T and TCR, and so we've wanted to start off with T cells and purifying T cells. And so as a great surrogate for a lymphodepleted patient, we actually sourced umbilical cord blood